Good morning, and thank you for the invitation to present some of our work. My name is Chris Van Hout, and I'm an investigator at the International Lab for Human Genome Research at the UNAM, Juriquilla campus. And give me just a moment to start my slides. And today I'll be talking about prospects for the genetic epidemiology of common disease at large scale in Mexico. Let me start by setting the context with UK Biobank, which really represents the state of the art in large-scale genomics. UK Biobank recruited 500,000 people through the National Health Service between 2006 and 2010, people in their 40s through 60s, who provided biological samples and agreed to have their health followed for 30 years. This power comes from linking this de-identified health records, cancer registries, imaging data collected at 22 centers across the UK. The breadth of data includes exome sequencing, whole genomes, metabolomics, proteomics, imaging, and specialized phenotyping. And the impact has been really enormous. In particular work that I led in a previous life in industry, we showed that gene burden analyses across 1,700 phenotypes have produced many validated findings, including some novel discoveries. And as one striking result, about 2% of the participants carry medically actionable genetic variants based on the American College of Medical Genetics guidelines, meaning knowledge of this genetic change has a clear and actionable clinical recommendation for those patients. Ultimately, UK Biobank may be the state of the art, but there are many biobanks and high quality and deeply resourced efforts to meta-analyze phenotype-genotype relationships are underway. Both for common variants, the Global Biobank Project is an effort to take advantage of large sample sizes to boost statistical power to detect variant associations, often with electronic health records-based phenotypes, generating genome-wide associations and phenome-wide associations, meaning a single variant with every phenotype for all available data. Similarly, for rare variant analysis using gene burden approaches, BRAVA, for example, is analyzing and making available these sorts of meta-analytical results at scale. But if you look closely at this map, most of these arrows point back to sampling in the north. Even the name here and placement of the arrows makes it look a little bit more global than it is. While harmonizing phenotypes between studies and performing the quality control that is required for meta-analysis of these studies is substantial, to say the least, the approach has been criticized as overly simplistic, focusing on additive genetic models, mostly in European ancestry studies, for easily obtained phenotypes of convenience. I jokingly refer to this as the meat grinder approach. Sequence goes in, numbers come out, turn the crank, not much thinking about biology. Of course, that's just the tip of the iceberg. Non-additivity, for example, may represent a minority of genetic effects, but are understudied. Environmental factors are often not very well characterized, and gene by environment and gene by gene interaction are secondary or tertiary considerations to identifying novel loci. Context or population-specific effects can be measured by the heterogeneity statistics in meta-analyses, but are rarely unpacked. And perhaps most relevant to this story today, traits are often considered in isolation rather than jointly, taking advantage of what we know from epidemiology and leading down a more integrative path. Here's an illustration of the diversity problem. This graph shows the ancestry composition of participants in genome-wide association studies up until mid-2025. And look at the colors. The vast majority shown here in red are of European ancestry. Latin American and Hispanic individuals shown in purple represent just 1.7% of genome-wide association study participants. Why does this matter? I can think of at least 3 reasons. First, genetic architecture varies across populations. Allele frequencies, effect sizes, linkage disequilibrium patterns all differ. Second, polygenic scores trained in Europeans show dramatically reduced performance in other ancestries. And third, we're missing population-specific signals entirely. This brings us to the Mexico City prospective study, which is really the focus of my work. MCPS is a collaboration between Oxford Population Health and the UNAM. The original aim was to understand how modifiable risk factors, things like diet, physical activity, alcohol consumption, smoking, all contribute to chronic disease of middle age. The recruitment was between 1998 and 2004, household to household in Mexico City, and recruited 159,517 individuals over the age of 35. Now, what's particularly interesting about that design is that recruitment was for entire households. From the exome sequence, we can estimate the proportion of alleles identical by descent for each pair of individuals, from which we count over 32,000 parent-offspring pairs and more than 30,000 sibling pairs. Very different than UK Biobank, for example, with counts in the hundreds. Today, MCPS is the single largest sequencing study of Latin Americans anywhere in the world. There's exome sequencing and array genotypes on over 141,000 individuals, whole genome sequence for 10,000 individuals, metabolomics data for more than 150,000 individuals, and there are mortality records for over 20 years. This, in addition to the baseline survey and a smaller resurvey conducted in 2015. So when I talk about MCPS enabling population health at large scale in Mexico, this is one of the resources that makes it possible. Let me walk you through where we're headed today. First is a description of the resource. I'll show you the MCPS cohort characteristics and the variant landscape we're working with. What does genetic variation look like in nearly 141,000 sequenced participants? Second, a statement of the problem. Body mass index has been the go-to measure for obesity-related risk, but I'll show you why it's insufficient as a proxy for metabolic health. There's important heterogeneity that BMI misses. Part 3 is our approach. We've integrated metabolomics data to capture that hidden metabolic variation, and I'll demonstrate how this substantially improves models of cancer mortality risk. And part 4 brings in the genetics. We have strong genetic signals for BMI in MCPS, and I'll discuss how we're working toward including polygenic risk scores alongside metabolomics in these integrated models. In terms of the demographics and clinical characteristics of the cohort, it skews about 2/3 female, mean age 51 years. This reflects the household-based recruitment. Diabetes prevalence is about 14%, over 21,000 individuals self-reported diabetes at baseline, and between 1% and 2% of ischemic heart disease, cerebrovascular events, and any cancer. About a quarter report any history of serious chronic disease. BMI is particularly relevant for what I'm going to show you. Median BMI in this cohort is 28.4, with an interquartile range spanning 25.6 to 31.7. So in this cohort, obesity is common. 78% are overweight with BMI greater than 25, 36% obese at BMI greater than 30. Now, two observations that impact our analysis strategy. First, when we look at mortality records, we can see substantial incident accrual. Cancer case counts, for example, often double when we include the mortality records. Comparing accrual rates to other resources from the Institute for Health Metrics and Evaluation, which draws from state and national level surveys in Mexico, we suspect substantial underreporting of cancer and other chronic disease in both the mortality and baseline records. This is important because it means we need to be careful about how we define disease status. For our consideration of cancers, we're specifically focusing on cancer mortality to avoid reverse causality. We need outcomes that come after our measurements of the metabolomics. I want to show you something important about disease ascertainment in this cohort using diabetes as an example. 21,000 people, about 14%, self-reported diabetes diagnosis at baseline. There are clear age and sex patterns, as you would expect, as you can see in this population pyramid. But here's the issue. Hemoglobin A1C is a biomarker for diabetes. It reflects average blood glucose over about 2 to 3 month period. So it's an objective measure. When we apply the clinical threshold of HbA1c greater than 6.5%, we identify substantially more cases than self-report alone captures. This tells us that there's meaningful underreporting of diabetes, either undiagnosed disease or reluctance to report. This isn't a problem with the cohort. It's a reality in population-based studies. But it matters for how we design our analysis. We use biomarkers to refine definitions when we can. And for cancer mortality, which is what I'll show you about today, we're using the death records as outcomes precisely because we suspect that they're more reliable than baseline self-report and because they definitively postdate the metabolic measures. So let's look at what we found when we analyzed the exome sequence from the MCPS cohort. This is work with Martin Velasquez from the UAC here in Querétaro. This is a site frequency spectrum showing the distribution of variants across different allele counts in our data set. What's immediately striking here, let me orient you to the graph. On the horizontal axis, we have the alternate allele count from alleles with only one alternate allele, 10, 100. So this is a log scale on the horizontal. And on the vertical, we have the number of sites at that alternate allele count, also on a log scale. These lines represent the different various types. Black represents all variants observed, blue, synonymous variants, this yellow or orange are missense variants, and red is loss of function variants. You'll notice that loss of function variants, shown here again in red, are particularly depleted at higher frequencies, which makes biological sense given their potential impact. And just to make this concrete, there's a table that shows the examples of each variant type. Working with over 92,000 MCPS participants who had their exome sequenced and were no closer than third-degree relatives to avoid the confounding of close related individuals, we identified approximately 11.4 million autosomal variants after quality control. And here's what really stands out. 5.1 million variants, that's about 45% of the total, were observed only once. We call these singletons. Approximately 11.2 million variants, representing 98% of all variants, are rare, meaning they have a frequency less than 1%. Now, you might notice these diagonal lines in the back. These represent the expectation under Wright-Fisher neutral model. That's a standard population genetics model, assuming constant population size and no selection. The relationship between the expected count of variants and the frequency is given by a pretty simple formula that relies on the effective population size, big N here, and the mutation rate, mu. But notice how our observed data deviates from this neutral expectation, particularly so for loss of function variance. This deviation tells us something important. These patterns reflect both recent population expansion in the Mexican and worldwide population, which creates an excess of rare variants. And in addition to that, with the loss of function variance, this represents ongoing purifying selection, which is actively removing deleterious loss of function variants from the population. The key takeaway here is that human exome sequence contains tremendous rare variation, and this variation is shaped by two forces: our recent population growth and the natural selection continuously working to remove harmful mutations. And this sets the stage for understanding why common versus rare variant analysis requires specialized statistical approaches, which we'll discuss later. So here's the outcome we're examining: mortality from BMI-related cancers. These are cancer types: kidney, liver, pancreatic, colorectal, esophageal, breast, endometrial, and others shown here, where there's an established evidence that higher BMI increases risk, though not all cancers have a linear risk. For example, the lowest risk in breast cancer is not the group with the lowest BMI. The risk relationship is more J-shaped. And though we'll use linear models to assess risk, I'll show you some graphics that address this potential for nonlinearity. In MCPS, during roughly 20 years of follow-up, 2,225 participants died from these cancers. That's 7.1% of all deaths, whereas overall mortality was about 20% of the total cohort. Now, here's the question. If BMI is associated with these cancers, does BMI alone capture the metabolic risk? The problem is that BMI is a crude measure. Two people with the same BMI can have very different metabolic health status, different lipid profiles, different glycemic control, different inflammatory markers, different fluid retention markers. So the question is, can we look at beyond BMI at the underlying metabolic state, and do these metabolic measures show independent associations with cancer mortality? This slide gets at the heart of why we need to look beyond BMI. What you're seeing here is the relationship between BMI and the metabolic markers in this Nightingale panel. This is a study conducted in MCPS on a subset of the available metabolic data at that time. Each data point in the circus plot is a different metabolite or biomarker. And in the red, you can see positive associations with BMI, and the lines in blue indicate a negative association with BMI. The metabolites are grouped by their metabolic class. So for lipid metabolism, and there are a lot of lipid measures in this panel, triglycerides, lots of measures of lipids from the standard LDL and HDL, apolipoprotein B. These are central to atherosclerosis and cardiovascular risk. Second, glycemic control markers as measures that reflect insulin resistance and long-term glycemic control. These tell us about diabetes risk and metabolic dysfunction. So HbA1c, glucose, lactate. Third, fluid balance markers, albumin and creatinine. And finally, inflammatory markers, glycoprotein A in particular. These capture chronic low-grade inflammation, which we know is linked to both metabolic disease and cancer. So we're not just measuring one thing, we're measuring distinct physiological axes that can vary independently. So yes, BMI is associated with almost all of these measures. That's not surprising. but BMI alone doesn't capture the full picture of someone's metabolic state. Now, I want to show you how we create an integrated measure of metabolic health. The methodology is pretty straightforward. And in work with undergraduates, Jair Contreras and Lorena Gutierrez, we use principal components on the metabolomics panel, including HbA1c. About 90% of the variance is captured by the first 10 principal components, 34% in PC1 alone. You can see this is the cumulative variance captured by each of the first 10 principal components. And in this slide, this figure next door, you can see the strong association of many of these metabolomics classes with their factor loadings on principal component 1. So when, as a group, when these metabolomics rise or fall, they tend to rise and fall together. We then model BMI as the outcome with these 10 principal components as predictors, in addition to some other age and sex variables. The fitted values from that model are what we call the predicted metabolic health, PMH. Let me explain the intuition here. PMH tells you, based on your lipid profile, your glycemic profile, your inflammatory markers, all the physiological axes we just discussed, what BMI would you predict based on those metabolic markers? If you compare that predicted value to someone's actual BMI, you can see the disconnect between their BMI and their actual metabolic state. If you look at this quadrant plot, the x-axis is PMH, the y-axis is the residual value. They sum to each individual's BMI. If metabolomics perfectly predicted BMI, everyone would fall on the diagonal. Clearly, they don't. In the upper right, high BMI, high PMH. These are individuals with obesity, with metabolically unhealthy profiles. Lower left, low BMI, low PMH, lean with healthy profiles. Those are expected. But if you look at the off diagonals in the upper left, these are metabolically healthy obese individuals, high BMI, but their biomarkers predict much lower BMI. These are individuals who are overweight, but metabolically protected. And in the lower right, metabolically unhealthy, thin individuals. Their BMI looks fine, but their metabolic profile is concerning. Some combination of dyslipidemia, poor glycemic control, inflammation, all of this despite normal weight. Heterogeneity is what BMI alone completely misses. PMH captures it, and we have this score for 151,000 participants. This slide shows the key result. Does metabolic health status, independently of BMI, associate with cancer mortality? We're looking at a logistic regression model. Cases are the 2,225 deaths from BMI-associated cancers, and controls are 147,333 individuals without cancer, BMI cancer mortality. The model includes a minimal set of established risk factors, age, sex, and BMI and PMH. The established risk factors all show expected associations. Age, being male, have positive associations with cancer mortality, just as we'd expect. BMI has a statistically significant positive association. Higher BMI, higher cancer mortality risk. But the critical finding is PMH. Even after adjusting for BMI and those other risk factors, PMH shows a significant positive association with cancer mortality. This is what I mean by independent association. PMH captures something about metabolic health status that BMI doesn't. Two people with the same BMI can have different PMH scores, and those with higher PMH, worse metabolic health, have elevated cancer mortality risk. Why does this matter? It means that we're not just rediscovering that obesity is a risk factor. We're showing that the underlying metabolic dysfunction captured by integrating lipids, glycemic control, and inflammation is independently associated with cancer mortality. Let's look at the distribution of these risks in different strata of BMI. This plot shows the prevalence for cancer mortality on the vertical axis with the 95% confidence interval of the prevalence in BMI deciles. So on the left, the lowest 10% of individuals by BMI. Our model indicated a statistically significant linear increase in risk. And now you can see the association is significant and in the expected direction, higher BMI, higher cancer mortality risk. The odds ratios here are modest. From the betas on the last slide, we're talking about odds ratio of 1.02 per unit change in BMI. BMI is clearly a risk factor, but the pattern here suggests more modest effects, and the 95% confidence intervals on the prevalences are all overlapping. Now, if we can compare this to PMH, when we look at metabolic health status, the effect sizes are substantially larger. From the model, the odds ratio is 1.05 per unit change in BMI. So the lower end is lower and the higher end is higher for PMH than for deciles of BMI. In addition, the 95% confidence intervals on the prevalences of cancer mortality are no longer overlapping. Additionally, we don't see any clear nonlinear or J-shaped relationship across all BMI associated cancers, as I alluded to earlier for breast cancer. This is the key point. Metabolic health status, which integrates information about lipids, glycemic control, fluid balance, and inflammation, has a greater effect size estimate of cancer mortality than body weight BMI alone. Two individuals with the same BMI can have very different cancer risks depending on their underlying metabolic state. Now, you might be wondering about the genetic component. Here's our GWAS for BMI on the full MCPS cohort using the Global Screening Array version 2, or about 350,000 variants after quality control. The result is well calibrated, so we have good statistical control over the close relatedness structure that I mentioned before, and population stratification, which is substantial in MCPS. As you can see, multiple genome-wide significant signals are apparent. These include some of the usual suspects for BMI, FTO, MC4R, the same loci that have been in dozens of studies worldwide. This suggests that the genetic architecture of BMI is at least superficially consistent across populations, and it is reassuring. But more importantly, the strong polygenic signal here suggests we can derive useful genetic risk scores from these data. Now, this is important because most polygenic scores for BMI have been estimated from European populations. When you apply them to an admixed or non-European population, they lose a substantial proportion of their predictive accuracy. With MCPS, we intend to build ancestry-appropriate scores that are more accurate in this Mexican population. And here's why that matters for our cancer story. Genetics captures your inherited predisposition. Metabolomics captures your current metabolic state, what's happening right now. And ultimately, these are complementary pieces of information. So our goal is to integrate all three, BMI as a basic phenotype, metabolomics for current metabolic health, and genetics for inherited risk, into a comprehensive model for predicting cancer mortality. In fact, GWAS and rare variant analysis for BMI and MCPS have already been characterized. So let me briefly acknowledge prior genetic work in this cohort. Again, this is a publication from my days in industry. Genetic associations for obesity and BMI-related traits in MCPS have been published, including meta-analyses of rare variant burden tests. This work identified genes including GPR75, where loss of function variants were shown associated with protection from obesity. You can see this in the plot in the lower left. Excuse me. Where the allele frequency, again on a log scale, is plotted versus the effect size. This observation in GPR75 is a unique example of a variant that has a high effect size but is protective. Loss of function is protective from obesity. This is a study of 3 different cohorts, one from the US, one from including Mexico City prospective study, and the UK Biobank. This observation is supported by a meta-analysis approach. You can see the distribution of the beta coefficients, the number of genotypes, the p-value in each individual cohort. And in addition to that, the combined meta-analysis result that indicated this association is genome-wide significant. I mentioned that this is a gene burden result. In this graph, you can see the exon structure of GPR75 and the distribution of the variants and their associations with BMI here in standard deviation units. This is driven by a handful of variants, but the approach is illustrated well in this graphic to combine variants at the level of a gene to test for association with rare variants, whereas individually, these rare variants would have very little power to detect any association with really any trait. So the prior work answered the question, which genes matter? Our work is answering the question, what's the aggregate genetic architecture? And can we use it for risk stratification alongside metabolic markers? So let me summarize what we've learned from this work. On the genomic side, we've characterized the genetic landscape in nearly 100,000 individuals of Mexican ancestry. 98% of variants are rare, which tells us something important about recent population history and ongoing natural selection. We've evaluated the genetic architecture for traits like BMI, suggesting we can build useful polygenic scores, and the extent to which polygenic scores are portable can be evaluated by contrasting European ancestry-based scores to those derived from MCPS in a trained test approach. On the metabolic side, we've demonstrated something crucial. BMI is inferior to the deeper picture of the metabolic profile. There's a substantial metabolic heterogeneity hidden within any BMI category. The predicted metabolic health score, which integrates metabolomics data, significantly outperforms BMI for predicting cancer mortality. Here, the key is integration. By combining genetics, which tells us about inherited predisposition, with metabolomics, which tells us about current metabolic state, alongside BMI, we can identify high-risk individuals that single-factor models would miss. So what is the impact of this work? MCPS is the largest multi-omic analysis in Latin America, which means that we can build risk models that work in this population. We've demonstrated that metabolomics substantially improves cancer risk prediction beyond BMI alone, and critically, this approach identifies higher risk individuals who might be missed by traditional BMI screening. In terms of ongoing work, we're now focused on extending this framework to diabetes, cardiovascular disease, and other morbidities associated with BMI, and working to integrate the polygenic scores with these metabolic predictions. Finally, and perhaps most importantly, this work is training Mexican researchers with access to world-class data and analytical tools, which is how we build a sustainable genomics capacity in the region. So finally, let me thank the people who made this work possible. Martin, Jair, Lorena, and Leo, all undergraduates who contributed their time and energy to develop these projects. I'd like to acknowledge my funding and thank you for your time and attention. And given the recorded format, please let me know via email on the screen if you have questions, comments, or you might want to be involved.